So this lecture was prepared by Ido Diamant, who is a bioinformatics software engineer. He is here on this lecture, and I will actually deliver the lecture, but if you have questions and you want to help with some of those steps in the lecture, Ido is the person to go. So the first part, so last class, we learned about Unix commands and some Python, getting started, basic variables and other type of constructs in Python. Today we're switching to Jupyter Notebooks, and Jupyter Notebooks are still a place where you can write Python code, but it's a nice environment where you can also do other things, and but it is a Python development environment. And I know that you guys have some background in MATLAB. So Jupyter Notebooks really take some of the elements that MATLAB provide. Initially it was MATLAB and Mathematica that had this type of notebook architecture for developing analysis workflows. And Jupyter Notebooks made it free, and it really exploded in the field of data science. And the idea is that you have the code, the Python code, also together with Markdown text, and we'll go over a little bit about Markdown. And also you can see the visual output of the notebook, a little bit like MATLAB. So Jupyter Notebooks, they have cells in them, and those cells can be either code, Markdown text, or the executed code. So it could be textual output or data visualizations. In the background, those notebooks are run with the IPython kernel, which we discussed last lecture. And you can get more information about Jupyter in that link, but Jupyter Notebook needs to be installed in your environment. So the first thing to do is to make sure that you have pip. In order to get pip, which is management for Python packages, you want to have pip so you can install different Python packages into your environment. And to do that, you want to type Python 3-m ensure pip. So if you can follow along and type that, and I assume some of you have it, but for those who don't, it will install pip for you. And then once you have pip installed, you can do pip install Jupyter on your command, and that will install Jupyter for you. And then once that's installed, you type in Jupyter Notebook, and then you should see this. It will invoke your browser and will create this environment that will allow you to navigate through your files and folders from the browser using the Jupyter interface. This is actually more difficult part. After you have that done, you should be able to follow along pretty well. So this is from the terminal. So you want to start the terminal, and then you want to type those 3 commands in order. So once you have the notebook, we want to create our first Jupyter notebook, and it's going to be, again, the hello world. So first, from the top menu, you want to select Python 3 IPython kernel notebook. So there are other languages that are supported by Jupyter Notebooks, but we are sticking with Python. And then you should be able to see this interface where you get a cell created. It's a code cell. So you can see in the drop-down that this is a code cell. And here you can start typing your code. So in this cell, if you can type print, open parentheses, quote, hello world, close parentheses, and then press run to see how this particular command is executed in the notebook. So now we're going to have a variable, and you can put some string in the variable, in quotes. And then if you type in message and then press run, you will see that the information is stored in the cell, in that variable over time. So as you go along, the value that you assign to the variable persists across cells. So message in general, in the beginning, it had this variable can be stored between cells. And then you can see that it has a string, and then we assign the number 2 to it. It takes that value 2, and then if we assign to it +1, it will produce the number 3. So initially it was a string, and then it was converted into an integer by the assignment of the number 2. So the whole point here is that it can preserve the values from cell to cell. So the value is getting stored. So if you guys can try to type those commands or this code and then test it to see that it works. So now we're going to add some other types of cells, not just code cells. This is the Markdown. And we're going to have a very quick and brief intro to Markdown. I hope many of you know like HTML. I'm assuming that some of you heard or seen Markdown before, but Markdown is just text that can be beautified and like HTML with tags. And here is some of the major tags that you need to know, it would be useful to know. So there is different tags for titles or subheadings that has this pound sign and then the title. You can have italics and bold, as well as numbered lists, and also obviously plain text. And you can also embed links and images into, then there is a whole guide for Markdown. And this is also very useful for other applications. We may face it again in the future in the course. But this is really a way to comment your code or reports with a nice text. So the next step is for you to go back to your notebook. You can create a cell. So you want to use maybe the plus sign. Create a cell and change it to Markdown instead of code. And then you can have put in some title. So you can have a pound sign and then give it some name. So this is the execution of the Markdown examples that we showed. So you can see that it's just have like different sizes of the header, and then you get italicized list, and then the alt for the image. There's no image there, but if there was an image, you would have seen the image. Now you can basically put all those things together and see how potentially this is kind of like building up into a report. You don't have to type in here or anything, but just get that idea of like you can mix up a Markdown, you can have some code, and then you can also have some images and the execution of the code, and you can see how this can become a report of a data analysis workflow that is well documented. Those notebooks, like right now, we are executing them locally, but you can run those notebooks also online. And there's some advantages of doing that. You can have computational resources that are more robust. So if you have to run some very big job, you may have to pay for it, but you can run it on large computers. And this can be done, for example, on Google Colab. You can also share your notebooks, store them. It's integrated with Google Drive. So the same way you share in documents using Google Docs, you can share your code and notebooks with Google Colab, and it saves it on your Google Drive. For low-end computing, like if you just want to test, for example, the notebook you just created, you can upload them to Drive or to Colab, and then you can see how you can execute those, and this is free. But once you try and do some more demanding jobs, you may have to pay a little bit, and it's not that expensive. So this is one place where you can execute your notebook online. Another place to execute a notebook is called Binder. And Binder takes the notebooks directly from GitHub. So the next lecture will be about GitHub. So you'll know more about how to use this sort of like central place where people are sharing and documenting their code, and it's like a version control system. So once you have a bunch of notebooks, you can put them on GitHub, and then execute them with a binder or my binder, which is more of an academic initiative for executing notebooks. So this is just another famous place to run your notebooks online. So our group historically was one of the first, or we were the first, to publish a paper in the form of a Jupyter notebook. So this was very new in 2016. And Zichen Wang was a graduate student in my lab. He wrote RNA-seq analysis pipeline that was published as a notebook. And this was specifically back then, Zika was a major concern, health concern, and he created a pipeline to analyze some Zika data. But the whole point was that the pipeline could be repurposed for any type of RNA-seq analysis. So a lot of people downloaded the notebook and used it for their RNA-seq analysis pipeline. Later on, Alex Lackman, who is here today, and he will give you some of the future lectures. And Dennis Torre created this resource called Biogroupies. So Biogroupies was sort of like the next generation Jupyter Notebook RNA-seq analysis pipeline that we created. And here you can come in with your FASTQ files or your gene expression table, and you can upload your data onto the cloud. And then it automatically generates RNA-seq analysis report in the form of a Jupyter notebook from your data after following some steps that you have to specify the metadata, the samples, select your control and perturbation samples, select the various tools that you want to use for your data analysis, and then it generates automatically Jupyter notebook that is executed in the cloud. And more recently, we created this next generation a notebook generator system, which is called Appyter. And Appyters are ways to convert Jupyter notebooks into web-based applications. And so far, we created about 100 Appyters that perform different types of data analyses. And all of those Appyters are served in this Appyters catalog. And we will learn more about Appyters today. And this is also part of your final project to build an Appyter. And you'll see that it's not that hard. You'll see that there is a lot of opportunity to create very useful tools for biologists using a very basic principles and code. And yeah, we'll go over it a little bit more. So just to summarize, we just learned how to install Jupyter notebooks. We wrote some code and it created our first Jupyter notebook. We saw a little bit about Markdown, combining Markdown with code, as well as learn about the two online platform, Google Colab and MyBinder, and some publications from our lab that use Jupyter notebooks to produce data analysis workflow for bioinformatics. Here are some resources to, obviously, like I said in the first lecture, you're not going to become an expert by taking this course. We just giving you the tools to get started. All of those resources that were mentioned are listed here. So you should take advantage of those resources, slides, and go to those websites and check them out and potentially take some tutorials.